Bismillahirrahmanirrahim. In the name of Allah, the most beneficent and the most merciful. Assalamu alaikum wa rahmatullahi wa barakatuhu. All of you, my dear students, how are you all? I hope you all are fine and well and enjoying your studies. Dear students, I welcome you in Pakistan International School TAIS virtual learning classes. And this class is for biology for grade 8 and which is said to be 9 junior class. In today's lecture, we will talk about cell cycle, some more things, and in that today we will see about the cell division because we have started cell cycle this chapter before in the previous lecture in which we have studied about um, that interphase stage, which is very important stage before mitotic division. So in the previous lecture that we have studied that before mitosis, cell prepares itself for the division. In that there are lots of changes that cell organelles duplicate and chromosomes duplicate or proteins, enzymes become more in number, and then we will now we will see the next step of that is mitosis. Mitosis comparatively very short duration uh, and as compared to interphase is the long we have studied that in cell cycle is the longest stage. So let's get started about mitosis. Mitosis actually is the cell division. So what is mitosis? You can see in the diagram that mitosis is actually the division of the cell from one cell into two cells with equal number of chromosomes, with the same number of chromosomes in both of the cells. How the cells divide and why its division is important, we will study in the next slides. So how it is done, first of all we will talk about this step because this is very important, that how mitosis is done and how the cell divides in our whole of the body, and it is going on in from the start of like before the birth up when this again sperm fertilized, so the division has been started, and up to the deaths the division is the like continuous process in every part of the cell of the body, sorry. So mitosis was discovered by a German biologist, the Walter Fleming, in 1880s. So you must remember the discoveries and the years and the names of the scientists. Yes, they can come in MCQs. So he saw some dividing cells under the microscopes. Uh, when he was observing the cells, so he saw that nucleus passes through a series of changes, which he said then mitosis. So that is actually mitosis. We can say that occurs in eukaryotic cells. In eukaryotic cells, like where all of the mm, like cells are having proper membrane bounded nucleus and other membrane bounded organelles in the cells, and in multicellular organisms that is done in somatic cells. Somatic cells undergo mitosis. Somatic cells, what are somatic cells? Actually, somatic cells is the all cells of your body, they are called somatic cells. So including the stem cells, these are the basic cells through which all the cells can form according to the conditions, like according to the situation, when the cell needs stem cells start to differentiate into other cells. Bone cells are also somatic cells, blood cells, muscle cells, fat cells, skin cells, nerve cells, endothelial like skin cells, and these are actually pancreatic cells and cancer cells. These are some all somatic cells. And these sex cells, which are talking about here, these are called germline cells. So germ cells or germ-like cells, they are said to be sperms in male and ovum in female. They are germ cells. Uh, there is no mitosis. Mitosis is done, but they go under the process that is called meiosis. First, they go under mitosis, definitely. Then they go under mitosis also. First mitosis, then they undergo meiosis, through which they form four haploid. I have told you before, haploid cells. Haploid means half number of chromosomes are there in these cells. From male sperms which unite with ovum female, so the fertilization occur and zygote will be formed, and after that by mitosis and mitosis again embryo has been formed. So these are germline cells, they undergo meiosis, and all the somatic cells or cells of the body undergo mitosis. So that is about prokaryotic cell. What about, eukaryotic cell? What about prokaryotic cells? They undergo a process that is called similar to mitosis, but not the proper mitosis. That is called binary fission. Binary means two, fission means breaking. So in binary fission, they do not undergo proper mitosis. Why? Because they don't have membrane-bound organelles, as well as they don't have centrioles in them. When they don't have centrioles in them, they don't have proper nucleus. That's why they don't form spindle fibers. So that's why they just duplicate their chromosomes, and chromosomes move apart on both of the side of the cells, and the cell break from the center, cytoplasm breaks from the center, and then this is called binary fission. That is very simple asexual division of the cells. Asexual, where the sex organs are not included, like sex cells, ovum, sperms are not included, just the cells, they don't have any type, so they just divide simply by binary fission. Binary means two, from bi, and fission means breaking, like the cells break into two cells where the chromosome number are same, but we can say that they don't go through mitotic process properly. That is done in prokaryotic cells, for example, in bacteria. So bacteria, archaea bacteria, they are prokaryotic cells, so they do go under binary fission, not mitotic properly. What are the stages of mitosis? The process of mitosis, actually, that is not a simple process. That is highly regulated and complex process. So it undergoes four main phases or four major steps. That first of all, two major steps. In two major steps, there are several subphases also. So it undergoes, first of all, through the division of nucleus, that is called karyokinesis. Karyo means nucleus, kinesis means breakdown. So it undergoes karyokinesis, like nuclear breakdown. And then it goes into cytokinesis, like cyto means cytoplasm, kinesis means breakdown. So undergo cytokinesis means breaking down of or division of nucleus, cytoplasm, first nucleus, then cytoplasm. So you can see here in this diagram that the cells, one cell that is going to be divided mitotically, so it undergoes the process that is called mitosis. So first this is interphase, early prophase, late prophase. So look, you can see this is nucleus because chromosomes are present in the nucleus. So first nucleus has to be divided, like chromosomes in the nucleus has to, have to be divided, and then the cytoplasm divides. So now we will see karyokinesis separately because it is the longest phase if we compare with the cytokinesis. So karyokinesis is the longer phase. Karyokinesis is called the division of nucleus, and it is further divided into 4 major steps. This is nuclear division. Among all this is nuclear division, that is called karyokinesis. Here there are 4 phases. Remember, is prophase, metaphase, anaphase, and telophase. Here you can see karyokinesis, like breaking off the nucleus, and then nucleus is reappeared again. So let's talk about these 4 steps one by one. Just listen and understand it carefully. These are very, very important steps in mitosis, and you will find this in every class, every upcoming grades. So you have to be very, very careful while understanding these concepts. Prophase, among all 4 phases, prophase is the longest phase because it is having lots of changing in the nucleus. So first of all, what is happened, the genetic material in the nucleus, we know that it is in the thin it is thread-like. So you can see here that is called chromatin. But after that, after duplication, this becomes the chromosomes, and 2 chromosomes. This whole chromosome is become this one here. We can see here, this is 1 chromosome. So 2 chromosomes just stick together to form 2 sister chromatids. This is 1 chromatid and this is other chromatid. So both of them joined with from the center. This is called centromere. So at the onset of prophase, chromatin condenses into highly ordered structure. These are called chromosomes. We know that they are duplicated in interphase stage, in S phase. They stick together in the center, that is called centromere. Here is very important thing that there is a protein in the centromere which joins these chromatids together. How can they join? Because both are all are DNA and protein. So they join together from the center by a protein that is called kinetochore. It is a complex protein structure, and here is the centromere, and centromere means centro means center, mere means molecules. There are some molecules of kinetochore by which they join together, and here in the center, centromere, there is a place where the spindle fibers will attach in the late prophase. Okay. So if we see here in the prophase more stages, these are the chromosomes, and you can see here these are sister chromatids, this one and the other 2, and this center is called a centromere. Centromere of both of the chromosomes, they stick together by this kinetochore. You can see here these proteins, these are called kinetochore proteins. So 2 centrioles now, they are present in the cell. We know the 2 centrioles. Okay, this you can see here, kinetochore protein, which further is used to attach with the spindle fibers. So we know that 2 centrioles are present, and these 2 centrioles are called These two centrioles are called centrosome. We have just talked about before in chapter number 4 that is just one centriole and this other centriole. They are actually in the pair which form the steric-like structure and they form spindle fibers. So one is called centrosome. There are two centrosome. Each centriole duplicates. So we know in that is duplicated in interphase stage, and then duplicates and thus daughter centrosome are formed. Like other copy of that is formed, and both centrosomes migrate to the opposite pole. You can see here on the opposite pole, and give rise to microtubules by joining tubulate protein present in the cytoplasm. We have studied about tubulin proteins because that is the part of cytoskeleton in the chapter number 4. So the protein is we have studied that in interphase stage microtubules or tubulin protein is duplicated, so that tubulin protein is used to form microtubules, and these microtubules thus form these spindle fibers with the help of the centrosome. So complete a set of spindle fiber, this whole complete set of spindle fibers, that is called mitotic spindle, because that is used for the process of mitosis. That's what is called mitotic spindles. So by this time now, this is a late prophase now, where nucleolus and nuclear envelope has degraded. You can see here is nuclear envelope with lots of nuclear pore. These are centrosomes. So these are two cells. Here you can see a complete prophase stage. So this has been duplicated and condensed, so they become chromosomes. And then these two will move, the centrosome will move on both of the sides, and spindle fibers has to be formed. So due to formation of spindle fibers in the late prophase, what happened, these nuclear membrane and nucleolus, this is nucleolus, so they will be disappeared, but they will reappear again in the telophase stage. So this is all about animal cell. What will happen in plant cell? We have studied that centrioles are only present in the animal cell. They are not present in the plant cell. So how the prophase will go on? So because they are highly vacuolated cells, so large vacuole is present over there. So before that, before mitosis, new vacuole has to be released its all fluid, and the nucleus has to be migrated in the center. When nucleus migrated in the center, then because a plant cell lack centrioles, so spindle fibers are formed by the aggregation of tubulin proteins. There are no centrioles, but like tubulin proteins are all around in the cytoplasm, so they aggregate and they form the spindle fibers over there, which are present near to the nuclear envelope. So nuclear envelope then disintegrate, and all the process is same like in animal cell. So you can see here the centrioles are moving on both of the sides, and this moves spindle fibers. It will form spindle fibers, fibers, all that is called mitotic spindle. So now we will see the next stage. Now before we have seen the longer stage of mitosis, that is prophase, in which spindle fibers are formed, centrioles are moved on the both of the sides, and we can say a nuclear envelope and nucleolus has been disappeared. Now this is metaphase. Spindle fibers have grown to the length. Now from here and here, both of the side spindle fibers are growing in the length for attaching the kinetochore. This is kinetochore of the chromosome. Kinetochore of the chromosome, because there are two, so from both of the sides, this is chromatid, and the chromatid, one will join from here and the other will join from the other side from this kinetochore. So kinetochore fibers, no, some spindle fibers known as kinetochore fibers, because they are attached with the kinetochore of the chromosome. And two kinetochore fibers from opposite poles, they will attach of the kinetochore of these chromatids, and chromosomes arrange themselves along the equator. Now they are going to arrange themselves on the line, on the central line, that is called equator of cell, forming a metaphase plate. Now, because they are arranged in the center, you can see here the spindle fibers are going to arrange and just joining, attaching with the kinetochore of this one. Then the number of other fibers are there. We can see now they are arranged in the center, that is called metaphase plate. You can see here metaphase plate, and in the center, and some other fibers are there like this whole is, that is non-kinetochore, because they are not attached with the kinetochore, but they are attached with each other from one centriole to another. So up to here, prophase and metaphase has been completed. Pro means first, phase means a stage, the first stage of mitosis. Meta is the prefix, it comes before the word, so it means that after. Meta means after. So like it comes after the first stage. Now ana also comes after the stage up to the last, like near about. Ana means like it is near to the end. So now what happens in anaphase? You can say metaphase plate is there. All chromosomes are attached here in the center. So now you can see the chromosome are moving on both of the sides due to shortening of these spindle fibers. When they contract, so they break from the centromere and moves towards both of the sides. So the pulling force divides the chromosomes, sister chromatin, this one and this one, both are sister chromatin, so break from the center, from the centromere, and moves towards both of the sides. So the other spindle fibers, non-kinetochore, also elongate. And after that, at the end, the anaphase cell has succeeded in separating identical copies of the chromosomes into two groups at the opposite poles. You can see here from the center, these fibers are just stretching these, and they are going to, they shrink from here, but they actually elongating and shrinking also. When they shrink, this side, they stretch and they stretch. So these break from the center and they move on both of the sides. Now the last but not the least, very important as well, telophase, that is actually reversal of prophase. How reversal of prophase? You can see here the new nuclear envelope forms here. You can see here contractile ring is formed. Now nucleus is going to break. Okay, nucleus has been broken down because it does not have nuclear envelope before. So it, both of the set of the chromosomes on both of the poles now. So at the end, what happens? Nuclear envelope forms around each of the separated chromosomes as it disappears in the prophase. That's what is reversal of prophase. So all the cell organelles again arrange themselves and reappear. Both sets of chromosomes now surrounded by new nuclear. You can see here, new nuclear envelope. So nuclear division is completed, but cell division has not been completed. One more step has left now to complete. So what is that? Yes, that is cytokinesis. Yes, cytokinesis, breaking of cytoplasm, is yet to be left. You can see here nucleus and nucleolus, nuclear membrane, all the organelles have been reappeared again, and the chromosome have become chromatin material now. So what is going to happen? One cell has to be divided into two. How the the cleavage furrow develops. You can see here the cleavage furrow. Like here, the cytoplasm, you can see it is coming down, and furrow is going to be. This is called cleavage furrow, and cleavage furrow where the metaphase plate, yes, it was in the center, so cleavage furrow will be developed in the center. The furrow deepens and eventually pinches the parent cell into two daughter cells. It's very easy in animal cell, but cytokinesis is sorry, here it is in not animal cell, here it is in plant cell. Cytoplasm in plant cell is a little complicated and different because there is cell wall. So cell wall has to be divided also. So we can see that several vesicles which are derived from Golgi apparatus move in the middle of the center. Why? Because Golgi apparatus vesicles are modified and they are having lysosomal quality. We know that it is having inside digestive enzymes, so they started to move towards the center. They move toward the center when they form a membrane bounded disc. You can see here all they appear in the center, and now they are going to form cell plate. This cell plate that becomes a cell plate, or that is also called phragmoplast because it is formed by the fragments of the Golgi apparatus. And first of all, it just appears in the fragments, and then it just straighten away and all the spread together. The plant grows outward. That is called, sorry, the plate grows outward and move vesicles fused with one another. Finally, membrane of cell plate fused with the plasma membrane here, and then it just deepens into the cell wall, and then resulting like slowly gradually releases its digestive enzymes, and this is going to break down from here. First of all, cell membrane, and then cell wall is going to be break down. Each bounded, the result is two daughter cells, each bounded by its own plasma membrane and cell wall, because this cytoplasm will release again its own cell membrane and then cell wall again around the cell. So plant cell is divided into two cells by the division of by dividing the cell by phragmoplast. So let's talk about the summary. You can see here the original form, which is taken by the electron microscope, how the cell is moving. You can see these are chromosomes are separating by the spindle fibers. Wow, it's amazing. It is going to be done every time in our body, in every cell. Okay, so you can see here number one step. So let's check it once again from a prophase stage. So here the cytokinesis, which we have talked about, but we'll start from prophase stage. So you can see here one cell, that is prophase. Chromatin is changed into chromatids and complete chromosomes. These are the pro-metaphase, like the start of metaphase. Spindle fibers are going to be formed and going to be elongated and is attached with the kinetochore. And here is metaphase, like the cells are in metaphase plate. Then in anaphase, the cells are pulling, the chromosomes are pulled by the contraction of the spindle fibers. And now in the telophase, nuclear membrane, nucleolus reappeared. and two nuclei are formed. Now what happened? Cytokinesis is done. Cleavage furrow, by cleavage furrow, two cells have been formed in this result. So, dear students, that's it for today. I hope you understood it. It is um, s- so interesting and so we can say amazing fact of the life, amazing fact of the life, like how our cells divide, how do we grow, our bones grow, like because you are in growing age, so mitotic process is very rapid in the childhood than in the teenager, but it is gradually slows down when the person gets aged. So that is very, very important indeed. So, dear students, I hope you understood and found it very informative. Wish you all the best. Inshallah, we'll continue in the next lecture the significance of mitosis. Allah Hafiz.